So I'm presenting a small project I'm working on together with Torbjörn, which is trying out a new method to answer the question how many fish there are in a river. So quite a basic question. And that new method is called genetic close-kin mark-recapture. So knowing how many individuals there are of a species or of a population is like one of the most basic things to know in monitoring programs or in conservation programs. Like in this example of a famous example where a cod population really drastically crashed, um, it's obviously important to know how many individuals there are. But knowing that, there can be many challenges. It can be, for example, that there are many individuals in a population, like in this school of fishes, or the individuals can be dispersed over very large areas, like this blue whale, or it can be difficult to observe the animals, like this camouflaged octopus. So normally it's not possible to simply count the individuals of a population. One has to do some methods to estimate how many there are. And the same is the case for salmon in rivers. As any animals in the water, it's kind of difficult to observe them directly. And still, it's possible to know how many fish there are in the rivers, for example, in relation to the construction of hydropower dams, to see if the population is doing good or if the population is decreasing. So there are different methods to estimate the number of fish in the river. This would be one method where people snorkel through the river and count the number of fishes observed, and then use that to estimate the total number of fishes. Or one can go through the river at night and use strong torches and to find the fishes and again count them directly. But these things are challenging in many ways. There can be bad visibility, water levels can be really high, that make it difficult, and that puts a lot of uncertainty into these estimates. So one classical approach is called catch mark recapture, where you take a sample of the population, you mark these individuals you have caught. In this case, you stain them orange. You can use any physical mark in this method. You put them back into the population and wait until they have mixed. And then you take a second sample and you count how many individuals are marked in that second sample. And then you can use that to estimate how many individuals there are in total. Like in this case, if there are 20 fish in total and you mark 8, you would expect it to find 2 of 5 in the second sample to be marked. While if the total population would be 40 fishes and you again mark 8, you expect that only 1 of 5 in your second sample are marked. So you can use this marking and the second capture and a count of the number of marked individuals to estimate population size. So this principle in this new method called close-kin mark recapture is the same. You mark the individuals, but you don't mark them physically, like staining them orange, but you mark them by catching their offspring and genotyping the offspring and the parents, and then do parentage testing between the offspring and the parents to find out how many parents have been marked. So because each offspring marks one mother and one father, you know how many fish you have marked. And then you take a sample of the adults and you look how many of these adults have any sons or daughters. In your first sample, you know how many have been marked and can estimate the population size. So in the most simple way, that would be done like this. If you capture 5 juveniles, potential offspring, you know that you have marked 10 adults. And then you capture 10 adults in this example and you check how many parent-offspring pairs are there. And if you have, for example, 5 detected, this straightforward leads to an estimate of 20 fish. So in reality, this can be a bit more complicated because the adults have, for example, because of different size, they have different fecundity, or each adult can have several offspring. So there are some things you have to do in reality in addition to this, but this is the principle. So we have heard a couple of things about salmon before today. To do this in salmon, you have to catch the adults which are migrating into the river, and you have to take samples and you have to release them alive so that they can have offspring in the river. And then you have to wait for the eggs to hatch, and you can take a sample of the offspring, and then you can do this estimate. And that's what we are trying in this project. We are using a population in this river, Victor, close to Trondheim. And the aim is then to develop closed-skin mark recapture for Atlantic salmon, to compare the estimates of that method to conventional counts, which have been done for Victor. And we're going to look at some other things also, like, for example, how many adults and juveniles do you have to sample, and in which way to get a reliable estimate of population size. So we needed adult samples, juvenile samples, to genotype them, and to do parentage testing. So some words about how that was done. Adults have been sampled in 2 ways in 2018. During conventional counts, some fish have been caught and scale samples have been collected. And a second method was that small fish have been marked with electronic tags, released in the river, and when they come back as adults, they are registered by this antenna in the river. And because samples of those fish have been collected, we know which adults came back, and we can genotype these adults without catching them. Juvenile samples have been collected the year afterwards by electrofishing, and in total 300 juveniles have been collected at 10 stations in the river, 30 fish at each station. So that's basically where we are right now. We are genotyping the samples, and when we get it back, we can do parentage analysis to check how many parent-offspring pairs there are, and can then work on the statistical methods to use closed-skin mark recapture. Thank you.